Hello and welcome to this presentation on the study titled Effect of the Depth of Straw Addition combined with Slurry Fertilization on Soil Microbial Community Composition and Respiration. There is little known how location of crop residues in the interaction with organic fertilization affects carbon and nitrogen transformation processes mediated by soil microbiota. So, we investigated effects of straw incorporation, slurry application, and their interactions on carbon and available nitrogen dynamics in the soil at different soil depths, microbial community composition determined by the abundance of bacteria, archaea, and fungi, as well as key players of the nitrogen cycle, and on activity of soil microbes measured by respiration. Soil used in the pot experiment was collected from the unfertilized grassland site in Ljubljana. The experiment was carried out in the open field under a canopy and lasted 4 months. There were 6 main treatments, as you can see on the right side of the slide, all carried out in 4 applications. Due to destructive soil sampling, 4 sets of pots per treatment were replicated 5 times, initially giving in total 120 pots. After setting up the experiment, we selected 24 pots per treatment, which were used for CO2 measurements throughout the experimentation. At the beginning of the experiment, the measurements were more concentrated to catch the initial flash of microbial activity. Thereafter, the frequency of measurements was decreased, as you can see on the slide. During the pot experiment, soil sampling for destructive analysis were done 5 times, as shown on the slide. The soils in these pots were sliced into 3 layers and used for laboratory analysis. Based on the soil respiration measurements during the experiment, we chose samples from 3rd, 13th and 49th sampling day to analyze the change in the soil microbial community. Among microbial analyses, we performed the quantitative real-time qPCR to measure the abundances of the total bacterial, cranarchaeal and fungal communities, as well as the abundances of functional microbial genes involved in nitrogen cycle. Among chemical analyses, we measured dissolved organic carbon and inorganic nitrogen forms. So, the results have shown that incorporation of straw into the soil induced higher soil respiration than surface-applied straw and control in the first 3 days of the experiment. Respiration rates in control remained the lowest throughout the experiment. Slurry addition increased soil respiration in all treatments, regardless of straw management. Dissolved organic carbon and inorganic nitrogen contents were increased in straw-amended treatments compared to control. In the straw-incorporated soil, the increase in dissolved organic carbon and nutrients was observed in all sampling depths at the initial phase of the experiment. These results suggested that with the enhanced decomposition, larger release of many nutrients and dissolved organic carbon from the straw-amended soils occurred especially in incorporated soil treatment, which might have stimulated microbial growth. Straw incorporation significantly increased abundance of fungal microbial community in the top 10 cm of soil compared to the control and surface-applied soil. This effect was even more pronounced in treatments with slurry fertilization. Among nitrifiers, archaeal and bacterial AMO-A gene abundances remained unaffected by straw management. Slurry increased ammonia-oxidizing bacteria, mostly in the 1 cm layer in all straw treatments. Among the nitrifiers, slurry stimulated near S, NOZ1 and NOZ2 genes with the greatest increase in the 1 cm soil layer. In the deeper soil layers, differences were not significant throughout the whole sampling period. Coupled effect of straw incorporation and slurry addition was beneficial for NOZ2 gene abundance in the top 10 cm layer. So, our results suggest that the combined effect of incorporation of wheat straw and slurry application may induce more active nitrogen cycling than just slurry application and crop residue removal. This could indicate improved nitrogen availability to the crops, which was also reflected in higher mineral nitrogen content in our experiment. Thank you for your attention.